My name is Dr. Daniel Charlebois, and I'm an assistant professor of biophysics at the University of Alberta. In our Ideas and Speculations article titled, "Does Transcriptional Heterogeneity Promote Genetic Drug Resistance?" we present ideas and speculate on whether non-genetic mechanisms may promote the development of permanent or genetic drug resistance. Antimicrobial resistance or drug resistance occurs when the effectiveness of a medication, such as an antibiotic, an antifungal drug, or other chemotherapeutic agent, is reduced. This is a major problem in healthcare settings around the world, with 700,000 people dying per year, and in a worst-case scenario, this number is projected to rise to as high as 10 million people per year dying from antimicrobial resistance by 2050. Non-genetic drug resistance arises through mechanisms that are not encoded in the genetic material of the cell, whereas genetic drug resistance occurs through a change in the DNA sequence or a mutation. We speculate that non-genetic mechanisms may promote the development of drug resistance in three ways. 1. Non-genetic mechanisms may increase the number of cells that survive initial drug treatment and then can go on to mutate. 2. Non-genetic mechanisms may increase the mutation rate inside the cell. And 3. Non-genetic mechanisms may increase the adaptive value of mutations under drug treatment. All biochemical reactions inside the cell are subject to fluctuations or noise. Part A of this figure shows that when a drug resistance gene is regulated by a positive feedback gene network, that these fluctuations are amplified. This is shown in the gene expression histogram, where the population in red has a high degree of cell-to-cell variability. In contrast, a negative feedback gene network suppresses these fluctuations. This is shown by the low noise population in blue in the gene expression histogram. Part B of this figure shows that when a high drug stress is applied, all the cells in the low noise or low variability population succumb to the effects of the drug, whereas a small fraction of cells in the high variability or high noise cell population survive this initial drug stress. ZOR is a gene that promotes resistance to the antibiotic zeaxanthin. Part A of this figure shows that random fluctuations in the expression of ZOR can result in the cell temporarily moving from a drug-susceptible state S to a non-genetically drug-resistant state R before succumbing to the effects of the antibiotic. Part B of this figure shows that when ZOR is modulated by a positive feedback gene network, that this non-genetically drug-resistant state is prolonged before the cell ultimately succumbs to the effects of the antibiotic zeaxanthin. Part C of this figure shows that if a mutation occurs while the cell is in the non-genetically drug-resistant state, that this mutation can lock in the drug-resistant phenotype. Many important unanswered questions remain in this field of research. First, though we now have evidence that non-genetic mechanisms play an important role in drug resistance in model systems, the prevalence of non-genetic drug resistance in pathogens remains unknown. Next, we lack studies that quantify the transition from non-genetic to genetic drug resistance. And lastly, if non-genetic mechanisms do in fact enhance the adaptive value of genetic mutations, what are the underlying mechanisms? Overall, evolutionary biologists and drug resistance researchers need to work together to develop a unified theory of evolution and to tackle antimicrobial resistance.